So we continue with introduction to cells and tissue culture. What is tissue culture? Basically, this explains when you see this figure right here. Okay, all of this is actually plants grown in media called agar. Okay, so basically this is coming from a big tree, but you cut it into small pieces and then you put it into the agar and you grow them until they get bigger. Okay, so there are three types of tissue culture: organ, tissue, and cell. So it depends on where what you want to put into. For example, organ such as seed, tissue, which is you cut it, and or maybe cell, which is you chop cells and you put chemicals to get them separated into cells. For tissue culture, the word right here, okay, if you see the word right here, totipotent means total potential. Okay, total potential means it has the total potential of becoming anything. Okay, so okay, totipotent. It has total potential. So when you say total potential or total potential, that means if let's say you have one cell that has not been differentiated yet, that means that cell can be anything. For example, it can be a root, it can be a leaf, it can be anything. Cell turns into root, leaf, branches or stems. So that is what it means by totipotent. It has a total potential of becoming anything. Okay, so it starts with the very small pieces of plants, or called explants, and we can produce disease-free clonal material. So from basically, if you take it from the tip, the meristems, they are disease-free. So you can culture them to produce a new plant. Okay, so cell culture, basically a tissue that from an explant it is dispersed. Dispersed means bersepa into cell suspension, which may then be cultured as a monolayer suspension culture. But usually this one is for animal culture. We can just go over past this. Why do we need cell culture? To look at cellular behavior. Okay, so it's okay. We're gonna pass this. For this one is usually for animal. So this is for basic tissue culture procedures. You can start from buds, roots, nodal segments, seeds, and then the next thing you need to do is to trim them, potong, and then you surface sterilize. Surface sterilize means the surface has to be cleaned, completely clean, and in this case we use 20. Most of the time, 20% sodium hypochlorite. Sodium hypochlorite is, for example, you take Clorox. So Clorox contains sodium hypochlorite as the active ingredient. So when you wash them, imagine that you are actually disinfecting your hands from getting infected with COVID-19. So you wash your hands all over. So in this case, you wash it with Clorox. And then you after you wash your plant, your explant with Clorox, you wash it with distilled water 3 times, and after that you dry them up and you put them into different culture tubes. So basically the small pieces are called explants. And you can get them from stem, from root, from petiole, hypocotyl, cotyledon, embryo, or meristem. So that's how you obtain explants. And the stages of micropropagation. Number one, the most important thing that you need, the first and the foremost, is you need to get the explant. Like I said, you can get it from the leaf, the petiole, the anything you cut. Let's say you have a big plant, you cut them down and you get the small explant, okay, the leaf, the whatever thing. Next thing, surface sterilize, which is you wash it with Clorox, you wash it with ethanol. This is such as the procedure when you are going out during the MCO. After you go home, you will wash your hands with hand sanitizers. So this is the same thing. You have to surface sterilize your culture before you put it into the medium. Next one, once after you have put them on the medium, we are going to proliferate. Proliferate here means multiply. So basically in that media you put cytokinin enriched media, and then you will see growth. But however, you will need to subculture at an interval of 4 weeks. Subculture here refers to something that you do usually, such as transplanting. But transplanting refers to working with the soil, but this one, subculture, refers to the activity that you do, changing media in the lab. Okay. And next one is rooting. So if you see that before this, you promote shoot formation. So in tissue culture, it is different from growing from seed. If seed, you will be growing. When you have seed, the things that will come out first is the root. But for tissue culture, the things that will come out first is the shoot. Okay, so make sure that you know that, and after you have the shoot coming out, then only the root will be we will do the rooting by applying auxin into the medium. And after you have root, you have shoot and everything, it's a complete plant. Next one, you transfer it to natural environment. Okay, so when you have after you transfer it, you will go through a process called hardening off, which is making them ready for the natural environment. So that means you will basically let them experience a more or subtle, or how do you say, we will not expose you to the natural environment right after you were cultured, but we will wait after you have grown bigger. Okay, so imagine that you are now a baby. We will not expose you to the environment, but we will wait until you are really ready, then only we will expose you to the environment. Alright, for the concept of totipotency, like I said before, totipotency refers to okay. Okay, the ability of cells such as an embryonic stem cell to differentiate into any type of body cell. Plant cells are also totipotent, which helps to explain why a graft of a plant can generate a whole new individual out of just a small branch cutting. For example, if you start with a small tissue samples from adult plant, okay, you start with tissue samples from adult plant, okay, and this is the pieces, and you grow them on the media, and they become callus, and you put hormones in the media, and vitamins and all, micronutrients, and eventually it will grow shoots, and you will have roots, and they acclimatize or hardening off, and eventually it will become a bigger tree. So the concept means when you start with a leaf plant tissue, which is basically nothing, okay, a cell that has not become a tree yet, but it can become a whole new individual of just a small branch cutting. So that means it has the ability to ability to differentiate into any type of body cell. So that's what is totipotency. Okay, so you can get from a plant, it can become a whole plant from root tip, single cells, callus, and also just from a single plant. So I'm gonna skip that. General lab requirements and general techniques. The things that you need for a tissue culture lab, one is the general washing area. So this is where you wash your stuff, your plant, your conical flask, and there is a place for you to prepare your media and sterilize the storage. Aseptic transfer area. So this one is basically referring to a place that is very clean, which has completely zero bacteria, fungus, and virus. So this is what it means by aseptic. It has to be very, very clean. Next one is environmentally controlled incubators or culture rooms. Once you're done with your activities, you need to keep all of your plants into the incubators, and also an observation or data collection area. So imagine that this is a lab, tissue culture lab. So this is the front, the outside. So you come in, and there's another room, another door. So imagine when you go into this door, it will produce a sound like psh. So this is how it works. So basically they are disinfecting your body, and you can probably see this during the COVID-19 that some of them are they have an ethanol shower, which disinfects the person who is trying to coming into the next room. Next one is laminar airflow hood, clean room. So this is a place where it is aseptic. Aseptic, as I said, aseptic means it is completely free of virus, bacteria, and fungi. And next one, after you are done with everything, you are going to transfer your stuff into the incubation or culture room, so which should be very clean as well. So these are the things that you need for in vitro propagation. So you can just read that. For water purifier, balance, and all. So these are next one. We're going to go to culture media, constituents, and preparation. So inside these are called media. So these are some of the media that we use in tissue culture. White medium, Murashige, Scoot, Gamborg, Shang, Anderson, Lloyd, and McCown. So the most famous, the ones that is mostly used is Murashige and Scoot. Okay, this is widely used in everywhere in the world. So the components of the culture medium, such as mineral elements, organic compounds, plant growth, regulators and hormones, auxins, cytokinins, gibberellins, and support system. Mineral elements, imagine that you are growing a plant in a regular environment. So you will put all of the fertilizers inside. Usually it contains NPK, for example, and it's same thing goes to culture medium. They also contain all of those elements that you need that are required for a plant to grow. And at the same time, you also add organic compounds such as charcoal. And you also put hormones such as auxins, cytokinins, and gibberellins in order to help them to grow root and shoot. And you also have support system. This one is to replace soil, which is in this case we use agar, because agar is hard, so it helps your plant to stand on its stem. The mineral elements, you have nitrogen, phosphorus, potassium, calcium, magnesium, and micronutrients are all these. So nitrogen is basically for protein building, phosphorus is for energy, potassium for signaling, calcium, and all those are for the health of a plant. Okay, so these are the sources. Okay, you can go over your notes. And okay, you can go over this in your notes. And next one is organic compounds such as sugars, vitamins, and myo-inositol. So basically, it helps to help you in osmotic potential. Vitamins is for carbohydrate metabolism, myo-inositol membrane cell wall development. And then you also have complex organics such as coconut water, yeast extract, potato extract. All these promotes general growth. And if you see right here, coconut water, actually they figured that it contains auxin, so that's why they put coconut water in the media. And also some people put activated charcoal to aid in root induction and prevent callus growth. Auxin is together with cytokinin, it affects cell elongation, advantages rooting. Okay, so this is the main factor of auxin. And also helps in callus induction, somatic embryogenesis, and there's several types of auxin. One is the natural one, and we also have synthetic auxins. The natural is IAA and IBA, but the synthetic auxins are naphthalene, acid, and 2,4-D. Meanwhile, for cytokinin, it helps in inhibiting embryogenesis, inhibit root induction, but most of the time it helps to promote shoot initiation. Okay, so this is the importance of cytokinin. So if you look at this graph, if you go down, the lower the amount of auxin, so you will promote shoot proliferation when you have a higher amount of cytokinin. But higher amount of auxin and low amount of cytokinin, you basically promote root formation. But at the same amount of auxin and cytokinin, you basically are promoting advantageous root formation from callus. And gibberellins promote stem elongation. Support system, like I said, this one is to something that replaces soil, and it helps your plant to stand on its stem. And you have agar, agarose, and gel, right? So all these you can imagine putting in your kitchen. So basically, that's how it looks like. It's the same thing. Next one is the effect of pH on cultures. Okay. The pH basically helps to the health of the media. Okay, it will not disrupt the function of a cell membrane, so it should not be too acidic, too basic. Most of the time, it's 5.5 to 5.8. Okay, so initiation of aseptic culture. Initiation means the starting. So for next one is initiation of aseptic culture. Initiation means the starting. How would you like to start and initiate your aseptic culture? Okay, so the things that you need in tissue culture is basically a very clean environment. So in order for you to prevent contamination, you have to have antibiotics in the media. That means when you are making the media, you have to put antibiotics and all of the drugs that you need in order to kill fungus or bacteria, anything like that. It has to have an improved lab condition. Everything has to be clean. You have to spray ethanol all over it. You have to sanitize. Everything has to be really, really clean. And work surface, it has to be clean as well. So you have to basically it all talks about cleanliness. And if working on the bench using a Bunsen flame, you need to heat the air surrounding the surrounding. Okay. For example, let me put some arrow here, ballpoint. So you need to swab all the bottle tops and necks with 70% ethanol Flame all bottlenecks and pipette by passing very quickly to the heart of the flame. So let's say this is your bottleneck, okay, and this is the cover. So you need to flame, okay, the side. Every time you open your bottle, it has to be flame on this side, so that if, let's say, any bacteria is flying and trying, or fungus trying to get into your media, it will be through with pressure. It will be taken out by pressure. It was sucked out by pressure. And avoid placing caps and pipettes down on the bench. Okay, so every time the same thing goes to your everyday activities. You should not put the bottles down on the bench, okay, because you want to practice cleanliness and clean the spills. Okay, sterile environment. So for autoclave, there is a machine called autoclave. I should show you. Okay, let's go autoclave. Okay, there is this machine. Looks like a washing machine. So if you put stuff in here, it will kill all of the things, contaminating materials, using pressure. So it's really awesome, and it's really murderous. Okay, so with a temperature of 121 degrees Celsius, 1 psi, 15 minutes. So everything should be killed. And if, let's say, if you're putting some materials that cannot withstand heat, so you should do ultrafiltration, meaning after you autoclave, you filter things into that media. Okay, so let's say you already have the media that has been autoclaved, so you cannot add other things that are not autoclaved. So in order for you to add sterilized medium, you have to filter them. So the size of the filter is 0.22 micron. And laminar airflow hood. Okay, I'm gonna show you laminar airflow hood. Okay. Okay, so this is basically you're working inside this machine right here, and where if every time you work in this machine, the air will be blown out from this machine. So that's why I put air flow hood. Okay. Next one is surface sterilization of explants. So every time you work with something, it has to be cleaned with ethanol. This is just like you working with something, or you try to clean yourself every time you go out to the market after during this MCO. Okay, so same thing. So you get take shower, put sanitizer, same thing. But this time around, you are going to use Clorox. And last one. Safety aspects of cell culture. So when you're doing tissue culture, it has to be very, very clean. So that's the biggest point in tissue culture: cleanliness and sterile environment. Alright, so that's it for today. I hope that everyone gets the point, and if you have any questions, just please ask me. See you next time in Google Classroom.